So our first speaker in this session is Jean-Pierre Changeux. I don't need to introduce Jean-Pierre, I think. Jean-Pierre was actually Stan's first collaborator, I think, although I think at the time he didn't know that was the case because you didn't even have a PhD. But yeah, so Jean-Pierre is a very famous neurobiologist, a huge contribution in characterizing protein structure before he turned into cognitive function, the development of the brain. And now he's going to talk about the global neuronal workspace. Thanks, Jean-Pierre.
Thank you very much. Thank you, Stanislas, for your invitation. I'm happy to be with you for this 60th anniversary. And also, I am very happy to remember all the good times we had together. And it was for me a real exceptional moment of science and friendship, which is not always the case. And I will speak today about the global neuronal workspace. Everybody knows about it. But I will emphasize from the molecular to the cognitive level. And I will split my presentation in two moments. First, the origins of the concept of the global neuronal workspace, and then how the molecular approach that we have been developing in recent years can be useful for cognitive scientists. That's my aim. So to start with, how did we met, Stanislas and myself? It happened that I was at a meeting organized by Edgar Morin in 1972, and I met Jacques Meller, who was a psycholinguist. And we discussed theory about learning by selection. We shall come back to that. And he was a very strong functionalist. I would say even an integrist of functionalism, up to the point where he had published a book in English where he asserted that it is not necessary to know the neural organization of the human brain to understand language. I jumped, as you can hear. There was a violent controversy between us, and we tried nevertheless, since we were friends, to find a solution. So the idea was to build common models with a common student. And he had two students in his lab. Stanislas was one of them, 20 years old. It's an anniversary, actually. He had read Neuronal Man, and with us the collaboration for more than 30 years. This is just to show you that Stanislas is a blonde. He is a blonde. You did not know that, but that's important. And this is the place where we have been thinking for these decades at the Pasteur Institute, at the same two seats. They are there, again, in my office at the Pasteur Institute. And of course, being in the Pasteur Institute, we were inspired by Pasteur himself. And quote, two sentences from Pasteur: "Without theory, practice is no more than a routine given by habit." And the second, "It's only by staying with the same studies for a long time that you can hope to see a little better than your predecessors." And I hope what I am going to say illustrates these two statements of Louis Pasteur. And our first scientific meeting in my office was in presence of the physicist, a very distinguished physicist from the Department of Physics at the École Normale Supérieure, Gérard Toulouse. And it happened that I met Gérard Toulouse at the summer school in Les Ouches, and he was also, like Jacques Meller, interested by the selective stabilization of synapse that you can see here, a model of epigenesis within, of course, limited genetic envelope. And the question is whether some of the models they have been using in physics could apply to the brain in general. And the idea was to use the spin glass theory, which is an advance in the statistical mechanics of disordered systems, and see to what extent it may apply to the brain. And the idea was that we start with an initial state, complex energy landscape with an abundance of valleys and peaks and so on, typical of spin glasses. And learning would then correspond to a gardening of this landscape, a pruning of the peaks and so on by selection. So this was, I think, an interesting conclusion, mostly because it contrasted with the model of Hopfield, some of you may have known, which is an instructive, I would say, Lamarckist model, packing of instruction from the environment. And I may say that there are still some people believing that, well, true or not. The conclusion was that spin glass supplies some kind of tree for synapse stabilization and elimination with a parsimony of genetic information. So that was a first attempt to approach global modeling of the brain. But I may say that I raise the question whether it's really interesting for us to keep going on that direction. And therefore, we developed together, and I think this is an important epistemological point for cognitive science, about what is a model. It is to represent a behavior or mental process on a minimal but realistic basis of neural architectures and activity distribution, molecules, in mathematical terms. Second, to build a formal organism. I like the word formal organism that we use, which is a network able to establish causal relationship between a specific behavior or even a subjective mental processes and objective neural molecular measurements. And last, this formal organism can be tested, and the best test, of course, is to ask the formal organism to pass the task. And therefore, for us, always in all our work, we started by beginning with a cognitive task and trying to build a model which is able to pass the task. And of course, all these models are produced by our brain, and every theorist should be aware that being a product of his brain, the best model is never give a complete and exhaustive description of reality. I think that's an important statement that we should not forget. So, well, this was quoted quite well. Now, what are the elements to use in the model? Of course, the neurons, the synapses, the millions of billions of synapses, the synaptic contacts, which link neurons together, and which are the key structure for communication between neurons. And now, progress of molecular biology have led to the identification of the receptors in the postsynaptic membrane here, even in solution after purification. And these molecules, which are known now at the atomic level, if I may say, are interesting because the channel which is opened by the neurotransmitter here, acetylcholine, is quite far distance from the binding site. So we have a communication between binding and opening. And the idea is that there is some conformational change of the protein which is taking place from a resting closed channel, open closed channel, and desensitize a high affinity closed channel, and perhaps other intermediate states that I've been discussing recently. Now, we had in mind this knowledge about the receptor, and also how one can build an interaction device between neurons based on synapses, based on the allosteric properties of the receptor. And very early on, with Stanislas, in a very early work, we proposed to make clusters of neurons which are interconnected by such kind of molecular device. So this has very few times being integrated into neuronal workspace. Most of those who are using artificial neurons simply forget that the neurons are made of molecules, which is a big miss. Having this in hand, we started to apply to some task which we propose to examine, which is learning by song, song learning by birds, and see to what extent one could have a model of song learning by selection. So the idea is that there is a critical stage in birdsong learning where there are 4 times more syllables than in the adult. Thus, the crystallization of the song would be some kind of syllabic attrition, a pruning of syllables. Question, does the pruning of syllables correspond to the pruning of synapses? And that was our aim, and these are the clusters. So we built a very simple-minded neural architecture with 3 layers, and most of all, spontaneous activity, and this is also often forgotten by cognitive scientists, that the brain is spontaneously active and produce pre-representation, what I call, and then application of a rule, a sequence to be learned, and have the elimination of the possible sequence through a mechanism at the time we call resonance with the input percept. So we achieved this goal, and the model works. And I may say that I am pleased to remember that it is consistent with our own view of the selective stabilization of synapse, but also by some work of Patricia Kuhl of syllabic pruning, and even a work of Stanislas on cortical representation of symbols, where objects and face prune back during early childhood. This is, of course, far-fetched interpretation, consistent with the language learning and cultural diversity in humans. So the next step was more elaborate in the sense that we tried to modelize the task with this Wisconsin card sorting task, where, as you know, subject is presented with a set of cards, which are different shape, colors, numbers, and he has response cards and should respond by following a rule, like, for example, when he has in mind red, then he should put the response card together with this card. And the doctor says yes or not. So it means that there is an intrinsic reward. I may say that yesterday, very few people, if not any, talk about reward. Even though in the training of monkeys, there are rewards. When you have a sequence of topics, you have rewards to build them, and so on. So this was, for us, an opportunity to try to modelize this aspect of the task and introduce level 1 with a sensory automatic, if I may say it, loop, and then a higher level which contains the rule coding and memory neurons, and where selection can take place by rewards. So this is, for me and for us, of course, a very important issue that one can have model which carry on such a cognitive task with these elements. And in addition, we added an auto-evaluation loop, which lead to something analogous to an reasoning process. We heard a lot about reasoning, but not, unfortunately, about auto-evaluation. But that's true. So we can check that learning takes place by selection. And most of all, there was a very interesting implementation that we propose, is that we developed a model of allosteric interaction between clusters of neurons, and a reward which stabilizes the one which corresponds to the rule and is accompanied by an allosteric transition, an allosteric switch, which is stabilizing the rule selected representation. In addition, I talked about the spontaneous activity in the previous model, and indeed we are here with a system working like the brain in a projective style. So the formal organism passes the task, and when lesions reproduce behaviors of frontal lobe patients. So that was a good step for us. And then Stanislas has been working with numbers, as you know, all of you. And we heard about that. And interestingly, we tried together to develop a model of infant numerosity detection. And there are two steps. One, to recognize the numerosity through numerosity detector. And then one, to compare the numerosity. And this comparison, of course, like in the case of the first case, involved a reward. But there is a memory which is playing a critical role here, and an auto-evaluation group, which is making the number comparison abilities with error detection. So that was a model of numerosity detection, which was consistent with the work by Andreas Nieder, the beautiful work he presented yesterday, and that is a good validation of this kind of theoretical modeling. So comes the global neuronal workspace. Then I was at a tragic moment, since in 92, as you know, Stanislas left. So our collaboration, maybe he was very happy with that, and it was okay for me, but that's what happened. And then I gave a course in 92 at the Collège de France on the neural basis of state of consciousness and attention. I mentioned Lamarck, sentiment intérieur, William James, the activity of cerebral hemispheres, which corresponds to it's a condition of state of consciousness. Linas and Paré, of course, on the thalamocortical oscillation and the differences between wakefulness, slow wave sleep, and REM sleep. And, of course, Bernard Barr book, The Global Workspace Theory, that I presented in my last lecture. And I was critical, very interested by it, but critical about it, because it's a psychological model, not a neurobiological model. So one has to make a difference between, if I may say, cognition and neurocognition. So we established this issue, and I also suggested that we could use the model that Stanislas and I has built on the Wisconsin card task. So Stanislas came back from the US. He was promoted chief of group in Saclay doing measuring, and in 1996 he came back to Pasteur. And we discussed about Barr's work, about the Wisconsin task, and he said, yes, keep going. All right. So that was an important step. Here it is. And the main idea, since this work was presented to you several times by Lionel and by Peter and others, I wish to stress one important point. What is really neuronal? And the idea is that, of course, to have a global physical workspace, we need to have interconnection in our brain at the brain scale. So, Caral had shown that there are a long-range connection, Dehaene and Patricia Goldman-Rakish, and more recently, the work of Van Essen, that in the cerebral cortex, 20% of the strongest connection links are consistent with this workspace, which link prefrontal, parieto-temporal, cingulate, and cerebellum. We did not put it in it. I put it in it intentionally with a question mark. And therefore, that was a proposal also that one can make the difference between conscious and subliminal simply by feed-forward presentation as Peter beautifully presented, and then global ignition when a threshold is overcome. So this modeling was used to pass a task, and the formal organism was imposed a very difficult task, which is the Stroop task. And in the Stroop task, you have words, but all the words are color words. And then you ask a subject, what is the color of this word? He said blue. Ah, I made a mistake. So I have to correct myself. And this is, of course, a very conscious task to correct and establish what is the right name that should be used. So this is a scheme, which is a connectomic scheme, which I think is very nice because it's simple-minded, but that it gives basic elements for this cognitive task. The processors of bars, the reward system, the vigilance system, the global workspace here, and the reward acting on the workspace for the selection of the right or bad word, and this give up to the naming response. Within the model, of course, we introduce the reward and this chemical help synapse that I already mentioned before, which is critical, of course, in the regulation of the workspace to establish the right solution. So then Stanislas went to continue his work on brain imaging, and on our side, we were more interested by the chemistry. And of course, immediately you realize that reward dopamine, vigilance, acetylcholine, and many other neurotransmitters is involved. So let's select a few experiments, like those done with the nicotinic receptor for acetylcholine. And here we discovered a long time ago that if you make mutation in the transmembrane domain here, then this mutation, instead of blocking the activity of the protein, enhance the activity of the protein, of the receptor, by about 100-fold. So these constitutive mutation can be interpreted through the model I showed with the different arrows by stabilizing by the mutation an active conformation of the receptor. Of course, this is at the origin of many diseases. Diseases with a loss of function are easy to correct. Diseases with a gain of function are much more difficult. It's a challenge from the pharmacological point of view. And interestingly, quite a few years ago, the group of Berkowitz in Australia discovered that some congenital autosomal dominant nocturnal frontal lobe epilepsy. Unfortunately, we did not have a talk about epilepsy by Lionel, but as you know, there is clearly a contribution of the workspace and the loss of workspace complexity taking place during the crisis. And here you have, in fact, a genetic determinism through a mutation, which is a constitutive mutation, exactly the one I described before. So this clearly illustrates that one can have mutation by gain of function of the nicotinic receptor, which is much more sensitive in these babies. And that, of course, there is a pharmacology here, and that one should be able to use nicotinic blockers to reverse the effect of the mutation. And I put also GABA, you shall see why. So this is for us an importance of allosteric receptor in disorders of conscious processing, of course. Now the question is whether we could use mice as a model. And of course, everybody knows Christoph Koch, Quest for Consciousness, Turing Task for Consciousness, and the distinction between a waiting period between stimuli and the absence of waiting period. If there is a waiting period, then there is an intermediate buffer, and according to him, it's a criteria, sometimes in a measure of consciousness, and therefore there is a difference between trace and delay conditioning. And I may say that not only it's valid in mice and rats, but also in Drosophila. That's what we showed quite a few recently, a few years ago with the group of Greenspan in UCSD. Now, he added if a single transmitter is critical for consciousness, it must be acetylcholine, and therefore the nicotinic receptor. Now, I have in the next few slides, I hope to be on time, a few experiments, selected experiments, done with the mouse. And here in humans, this is the work of Moutard and colleagues, that there are spontaneous resting fluctuations in the human brain, which are characteristic of his conscious state, which is recorded by EEG. Question, can we record these slow wave fluctuation in the mice? And the response is yes, using calcium imaging. You see here the awake state of the mouse with this spontaneous resting fluctuation. Anesthesia is there perturbed. And if you knock out the nicotinic receptor, complete mess. So there is also the possibility to use a drug, mecamylamine, which is a quantitative blocker, which perturbs this activity. Now, last but not least, with these mice, we can look at, and this is the work of Heinemann, essentially, the gene, the mutated gene, which confers the epilepsy I mentioned before, abnormal EEG, seizures, cognitive deficits. And the most interesting thing is that there is a 20-fold increase in nicotine IPSP, which means that there is an enhanced effect of nicotine on receptors which are on interneurons, inhibitory interneurons. So that's, for us, a quite interesting thing, since we are in a situation where always one considers activation for consciousness, but here it introduces a very interesting parameter, which is the fact that some of the receptors, in particular the nicotinic receptor, may act on inhibitory neuron, which themselves, if they make a chain, so either inhibit or stimulate the pyramidal cells involved in the conscious processing. I will close by 3 slides. They concern schizophrenia. And of course, schizophrenia is a complex disease, difficult to define. Well, I trust cognitive tasks, which were done by Charles and Dehaene in 2017, which is based on the monitoring of task deficit, conscious conflict, conscious errors. And they are clearly different in schizophrenic patients. Now, brain imaging shows an hypofrontality, which is, again, consistent with the global neuronal workspace without being really a proof. Now, the interesting aspect is that there are genetic predisposition to schizophrenia in humans. And here, the alpha-5, alpha-3, beta-4 subunit genes are deficits caused by mutation of the nicotinic receptor, and which cause alteration in the mice of the behaviors when you have transplanted this human gene into the mice. And therefore, you have a mice with a phenotype which is related. I don't want to say that they are schizophrenic mice, but they have a phenotype which is good enough to make the difference with normal mice. And on these mice, we have shown that we have, when we introduce the alpha-5 SNP, then there is an altered response to acetylcholine, which is taking place at their level. I am very short of those who are interested can look at the picture. And then the very interesting positive allosteric modulator of alpha-5 nicotinic receptor, galantamine, was used to restore the mutated response. Here is a deficit, stabilize the resting state, and then galantamine stabilize the active state. So we can restore the effect of the mutation. And very interestingly, we are here in the presence of a drug, which is FDA approved, and which is described in this paper on psychiatry research. So it means that we are reaching a level of precision pharmacology of the global neuronal workspace. So this is, to close, take-home message. 35 years of efficient and enthusiastic collaboration. This was in Leiden, when several neuronal models of cognitive functions, including I did not talk about the Tower of London and many others. MENATCOM is a global neuronal workspace. And also, I would insist that from the origin, there was an appropriation of the molecular level in cognition. So we think we are now in a new field, which is molecular cognition, you like it or not. And there are many diseases which can be understood in terms of the global neuronal workspace. This is in progress. And a new allosteric precision pharmacology, as I mentioned before, is to develop in the future. Happy birthday, Stanislas. Thank you very much.